Hi, my name is Laura Newcomb, and I am a graduate student in Emily Carrington's lab. And I've been one of the many people working on this question of how does mussel attachment change with changes in water temperature and acidification. So I just got back from the field yesterday, where in addition to pulling mussels to look at their attachment, I brought some home to do some dissections on them, which is laid out here. So I've taken the tissue out of the mussel so we can measure how much tissue is in these mussels to get a sense of their health. So I can show you a few things in the lab that we use both in the lab and in the field to get at this question. So the first here is a handheld force gauge that can measure the force to rip an entire mussel out of its bed on the shore or off of an aquaculture line. So we attach the mussel through the hook and pull up to record force. Sometimes though we want to look at the level of an individual thread. We have a different measurement device to do that. So this lives over here, and this is a force transducer mounted on a frame, so we can move it up and down to measure how strong individual threads are. And we have a bit of a fancier one up in Friday Harbor if we want to really look at it in more detail. That's a few things we have in the lab, and I'm excited to share some more of this research with you in the following talk.
This presentation is looking at the effects of elevated temperature and PCO2 on mussel attachment strength in a field and laboratory setting. So before I go on with the presentation, I want to explain a bit about the units or ways that we show changes in ocean acidification, and that is with PCO2 and pH. So ocean acidification is the process by which atmospheric CO2 is absorbed in the ocean, and we can measure that absorption by using PCO2, the partial pressure of carbon dioxide. Once CO2 is in the ocean, it reacts with water to form carbonic acid, and that product results in a decrease in pH. So ocean acidification that I show in the following slides is shown both as an increase in PCO2 and a decrease in pH. And to put these values into context, in the open ocean, the average PCO2 is 400 microatmospheres, and the average pH is 8.1. So you go to any rocky intertidal shore, you'll be certain to see mussels all over this shore, as this photograph shows. And they're quite iconic, both in their presence and their importance in holding down and structuring these communities. Mussels are also important economically, and mussel aquaculture uses a mussel's ability to self-attach by growing mussels on lines showed here. So on the left is a mussel raft, and this is in Penn Cove, part of Penn Cove Shellfish on Whidbey Island. And on the right is a picture that's looking underwater at these long lines of mussels growing. This is a photograph back on a rocky shore of a mussel bed. What we see is when this exact spot is photographed 2 weeks later, about half of the mussels have disappeared. And so what we see is that mussel fall off is happening on these shores, and that's creating these large patches. Similarly, we see fall off from aquaculture lines. So this line is nice and full of mussels, and that's what you would want to bring up if you were trying to harvest these. But sometimes lines come up like this, where there's large patches of mussels missing. So the farmers actually invented these mussel discs that you can see up on that left picture, and those help stabilize the columns and increase the chances that fall off won't occur. Yet sometimes even with these discs, they bring up bare lines. So to think about what's causing fall off, we need to look deeper into how mussels actually attach. So they attach with stretchy structures known as Byssal threads. So in this cartoon version of the mussel, the Byssal threads are coming out and attaching it to a substrate. And there are 3 important regions on these threads that are functionally and structurally different. So at the top, nearest to the mussel, is a stretchy proximal region. Next is a strong and stretchy distal region. And last is the adhesive plaque, which serves as the glue to attach it to something such as another mussel, a rock, or an aquaculture line. So there's seasonal patterns and attachment strength. So here, going out every month and pulling mussels off the rocks, you can measure their tenacity, which is how strongly attached they are. And so weak tenacities mean that there may be more chance that they'll fall off, and being strong increases your chances of staying on. And what we see is that at the late summer, early fall, mussels are at their weakest, and this corresponds to the time that the greatest fall off occurs. However, we don't know what's causing this weakening. And so the question I've come to address is what physical factors account for byssus weakening. So the first thing that the lab tested was elevated PCO2, or low pH, associated with ocean acidification, and found that mussels exposed to these conditions over 3 weeks showed a reduction in individual thread strength at pHs beyond 7.6, below 7.6, and PCO2s beyond 1200. And the part of this thread that was affected was this adhesive plaque. So something was changing in the glue to make them weaker and popping off at lower strengths. We then exposed mussels to elevated temperatures. And what we found was that at temperatures above 18 degrees Celsius, we also saw a decrease in thread strength. And this was due to a decrease in the strength of the proximal region and the strength of the plaque. So the next question was, now that we know that temperature and pH individually affect threads, how do they combine to affect thread strength? So to think about this question, remember that there's 3 regions of the thread, and for temperature and PCO2, 2 of these regions, the proximal and the plaque regions, are the weakest links of the thread. And what we see is that PCO2 weakens plaques by making them twice as weak at high PCO2s than low PCO2s, and that temperature makes the proximal regions 3 times as weak at high temperature and the plaques 2 times as weak. And so with this framework, we approached this experiment, and we did this in Friday Harbor Laboratory's Ocean Acidification Environmental Laboratory. And here we exposed mussels to 3 temperatures and 4 PCO2s over 3 days. And so each of these coolers here is an individual setup where we manipulated the temperature and the PCO2 of the water. And after 3 days, we took the mussels out and tested the strength of their threads. And what we found is that we recovered that at temperatures above 18, we see a decrease in thread strength. And from low to high PCO2, so the low PCO2 is in the light gray and the high is in the dark gray, we saw at the lower 2 temperatures a difference between these 2 PCO2s in that we actually saw an increase in strength. So to think about these results, kind of an overall, is that under low temperatures, we see a PCO2 effect, but under high temperatures, we don't. So you can think about a mussel hanging on a rock, like a rock climber hanging off of a cliff. So, like a mussel, a rock climber has 2 points of weakness. Either he anchors in the rock in a weak place and can fall, or he doesn't anchor properly in a harness and his harness can break. And this is analogous to the mussel with its plaque on the rock and its proximal region like its harness. And so once one of these regions weakens, it doesn't matter what happens to the other because the rock climber or the mussel has already fallen. So what we see is that the temperature effect is much larger on the proximal region, and they break before we can see a PCO2 effect at that high temperature. So looking back at these results, we do see an increase in strength of PCO2, where the previous study I showed a few slides earlier saw a decrease in strength of PCO2. And one working theory we have is that mussels were exposed to these conditions for different time periods. So when they saw a decrease in strength with increased PCO2, that was over a longer term 3-week exposure. When they saw an increase in strength, that was a 3-day exposure. So maybe other factors that we didn't quite account for, such as time, may also influence this PCO2 response. So to conclude what we found from these lab studies, when we see strong attachment, that can be due to low temperature, short-term high PCO2, or long-term low PCO2. But when we see this weak attachment where many mussels are falling off of the lines, that may be due to high temperature, short-term low PCO2, or long-term high PCO2. And our next question was, do we actually observe these conditions in a mussel's natural environment? So to do this, we went to Penn Cove Shellfish in Penn Cove on Whidbey Island. So you can see Seattle is south of Whidbey Island. And if you fly over Penn Cove, you'll see these rectangles in the water. And what these are mussel rafts. And if you look under the water, as I showed earlier, you see these long lines of mussels hanging. And we went here and measured water conditions. So if you think about a mussel line hanging vertically in the water, we wanted to know, is there a difference between what the mussels at the top of the line and the bottom of the line see, and what parts are reaching over those temperature and pH thresholds that we've noticed in our lab studies. So for pH, which is plotted here, that 1 meter top of the line in black and the 7 meter bottom of the line in blue, we see two things. The first is that pH can vary over the course of a day or months, and we see differences between 1 and 7 meters. Our threshold line of 7.6 is marked in purple, and what we see is that especially at 7 meters, we dip below this threshold a lot. So yes, we do see pHs below the threshold that can weaken mussels. For temperature, our threshold is marked in red, and again, we see differences in temperature between the two depths. And while the daily averages here don't cross that 18 degrees, some of the hours on the day do reach above that. So we get very close to that temperature threshold. So next was to link together what's happening in the field with temperature pH to what's happening with the mussels living there. So we asked, how do differences in temperature and pH with depth affect mussels living at different depths along the aquaculture rope? So to do this, I went onto the harvesting boat shown here, and as they pull up these mussel lines, I took mussels growing at the top meter and the bottom meter and tested how strong individual mussels were. And these results are shown here. So low tenacities are weaker attachments and stronger tenacities is a stronger attachment. And this is data from August 2013 to April 2015 at both 1 meter and 7 meters. So if you look at the black line, we see a seasonal pattern where they're stronger in the winter and weaker in the summer. At 7 meters, we don't see as defined of a pattern with those huge differences in strength between winter and summer, but we do see that it's, first of all, not always the same as 1 meter, suggesting some of these differences in water conditions over those depths may change the attachment strength. So we've been now looking to link these changes seen in tenacity with things such as temperature and pH. And this part of the project is still underway, but one pattern we've seen emerging is if we plot monthly tenacity versus monthly maximum temperature, the months where temperature is highest are also the months when their tenacity, their attachment strength, is the lowest. And we hope to be able to pull this apart with other factors such as pH and to look greater at the 7 meter lines to see what's going on there. So our conclusions so far, in the lab, we've seen that elevated temperature and low pH weaken mussel attachment. In the field, we know we reach the temperature and pH thresholds. Additionally, in the field, we can see that at 1 meter, weak mussel attachment correlates with warmer temperatures. And what we're still trying to understand is that temperature and pH don't explain all the patterns, and so pulling out what other things may be causing these changes. So with that, I have a number of acknowledgements of both people listed on top and sources of funding on the bottom that have made this work and projects possible.